And it's a really exciting time, I think, to be talking about these topics. And I brought you today actually a whole host of really exciting projects of the recent past. And the title of this presentation is going to be Working Smarter, Not Harder in Drug Discovery, and using AI assistance to accelerate biological discovery, which I think is one of the most potentially high-impact areas that we can work on today with AI in drug discovery. Quick word on our team. Our team is called the Biomedical AI Group, part of GSK, a big pharmaceutical company doing drug development in a number of therapeutic areas. We are based mostly somewhere in Europe, different locations, but really trying to advance the science of AI in health and medicine, and really trying to use these AI tools to help us identify, monitor, treat people ultimately better using these tools. You see some of these faces, so all of this work really requires an army to get right, and we have fortunate support by all of these talented people to make these ideas reality. And I just wanted to start off with maybe a big picture view before we dive into some of the science. And one thing I always like to talk about is why we're doing it. Why is AI for drug discovery and drug discovery in general so important for the world? And I brought you the two plots that I think illustrate this, in my opinion, the best. The one on the left-hand side that you can see here, it's really a timeline from 1940s to all the way to roughly today, 2020s. And the chart shows you the FDA-approved medicines for lung cancer. And if you look at 1940, there was actually just a single option that you had as a patient with lung cancer. And quite frankly, this was not so much a medicine as a repurposed chemical warfare agent that we used to try to help these people. Now, fast forward to today, 2020s, you can see the vast amount of options that you have as a patient with lung cancer, some of which extremely targeted. For example, here targeting a very specific mutation in your cancer. So over time, we're offering much more availability and much more tools for you as a patient if you have lung cancer today to get better outcomes. Now, what we also want to see, and that's why it's so important to see the second-hand plot here, which is, again, same timeline. So we're looking roughly 1960 here, all the way to roughly modern age. And this is a plot of the survival rate that you have also in lung cancer. And you can see here, in the past, you were pretty much more likely to die from cancer in the next 5 years than to survive. But if we fast forward to today, we're looking at 70%-ish survival rates in lung cancer. So you're much more likely to survive this disease for the next 5 years than you were in the past. So this coincides with the development of these new effective options, is also an improvement in the outcomes that we see for these patients. So drug discovery and medicines, they do a lot of benefit for society, and they really help us address these grave diseases in a very effective manner when all things go right. The other side of the coin that I always like to talk about as well is actually we're doing so, yes, but a lot of these efforts have been quite slowed down over the last few decades. In fact, we're actually investing exponentially more resources in drug discovery than we have been in the 1940s. So we have maybe hundreds of times, maybe thousands of times more PhDs. We have maybe much more researchers in the world than in the past, but we don't have hundreds of times of more medicines. So why is that the case? And there's a really nice concept that is called Room's law, which you may have heard already if you saw Andreas Bender earlier in this session, where basically the idea is that the productivity in drug discovery is exponentially slowing down. Why is it called Room's law? It's basically the inverse of Moore's law that you heard in CPUs, right? So in Moore's law, we're getting the improvement. In Room's law, we're getting the decrease. And this is a quite grave problem because we're actually spending now somewhere around $1 billion. Different estimates. This is a bit of a science as well, but we're talking about billions nowadays to get a single medicine to market. Imagine that. There's not many projects in the world where you need $1 billion to get started. The other problem that we have in this field now is that the success rates are historically quite low. We're talking about 5 to 10% depending on which disease area you're trying to go after. This is also quite notable because it's, I think, one of the few domains in the world where failure is the norm. So there's very few industries where you go to and actually most of your things fail, right? This is actually very unique in the world that you have an industry where this is the case. So this is one of the hardest areas, I would say, to work on, and there's a lot of factors that make it even harder over time. Now there is some silver lining on the horizon. You can see here this red part that I'm showing, which kind of shows a break with the trend. It is a good thing. And people are attributing this breakthrough to the trend with the increase in personalization and the increasing availability of genetic data. And I will talk a little bit about how we can leverage these new emerging data sets and types to really maybe advance and really change this paradigm in the future. Now, taking a look at what works in this domain, right? So you might want to ask yourself, you know, if we're doing drug discovery for such a long time, what do we know works? What have we seen to be working? And you might be surprised there's actually only really two things that are solidly established to be working in drug discovery. One is population scale biobanks. There's pretty good science around drugs that are developed against mechanisms that are validated in large, big cohorts using natural variation of the genome. You can actually see that there's an enrichment of drugs to be successful if you're using that type of data. So understanding the mechanism, having causality behind what you're doing is one of the things that we know to be established, and this has been replicated a few times. The other thing that we know to be effective is actually called biomarkers, is actually knowing what to target in which types of people. And so I'm showing here the different layers of what is behind a single person's individual profiles, all the different types of data that may go into characterizing an individual's differences from other people. And so if you're able to find and pinpoint which types of people benefit from which type of modulation using your causality, you might be able to really enrich your drug success as well. And this is also connected in multiple studies from an improvement from around 2 times better success rate in immune diseases all the way up to 8-fold higher success rates if you're going into oncology. So really, if you take both of these elements that we know to be successful in drug discovery and you try to abstract them away, what's kind of the learning that we take from it? I would say it's having a causal and targeted link. So knowing what to target in which type of people and having a mechanistic understanding why you're doing so is really the one thing that we know to be working in drug discovery. Quite crazy, we're doing this for decades, but really there's only 2 things that we would say is proven to be successful. Now, really, the step back and the question that we will be talking about today, and really what I want to bring you today is how we can utilize AI to bring this type of targeted medicines with strong causal evidence to patients faster than we have in the past in order to accelerate this kind of development and give us more of these effective medicines, hopefully better than in the past. And quick agenda of the rundown of the session will be, so we just talked about why we're doing this, why it's important, but also I will tell you a few examples of how you can utilize these principles to really accelerate drug discovery. One thing I will be talking about first is using computers to do experiments, so using in silico equivalents of cells, of humans, of tissues, to really understand in computers before you go into the real world what might be happening. Secondly, we developed a generative model of biological mechanisms that can give you kind of a causal bottom-up understanding of what's going on in humans, and you can use that to understand what to go after. Another thing that we're doing is using optimal experimentation design, so really understanding which types of experiments, once you have this understanding, where do you go for in terms of validation. And then lastly, we also have quite a bit of work around doing causal inference, so understanding what is the actual underlying mechanism of observations using these different types of experimental data sets. And this is what I call the AI-driven stack for accelerated biological discovery. And so taking a little bit of a look at drug discovery in general, this is kind of an outline of the process. So if you're working at a big pharmaceutical company, this is basically your bread and butter. So you're starting here usually in the discovery stages, and this is what you will find most science on, right? So if you see a scientist or early stage research, early companies, biotech, startups, etc., they're usually operating here on this early stages. And this can take somewhere around 2 to 3 years. And usually what you're doing is you're looking at thousands of different opportunities, right? You're serving a big space, you're trying to find out what am I going after, and you're using these types of models and machine learning to really help you pinpoint the ones that can make it to the next stages. What then usually happens is you select certain of these candidates, of these ideas, to go forward in what we call the preclinical phase. This is usually where we do these experiments that are not in humans. That means cells, that means dishes, that means potentially animals in some cases. So really these early stage experiments that are not quite yet human data. Once you've done that, and once that is successful, it is successful roughly 60% of the time, you can go over to the phase 1 study. And the phase 1 study is really the first time you test your idea in a real human. And that is usually the point where a lot of projects then fail because a lot of the data and a lot of the prior experiments are not really meaningful for humans. So most of the ideas that were in the preclinical stage, they actually also ultimately don't make it. So we're having this kind of exponential drop off here. Success rate around this phase 1 is around 50%, so half of your ideas will not make it further. Once you're in that phase 1 and it works, and you show that your drug is somewhat safe to go to the next phases, you go into larger studies, and this is our phase 2, phase 3 studies, and usually there's very few ideas. Even for the largest companies in the world, only dozens maximum of ideas make it ever to this stage. So we're looking at maybe 5 out of the initial 10,000 that make it to phase 2, and the success rates plummet here, because then it's not only about is this drug safe, but is it working? Is it effective? Is it actually changing something that matters? And for most of these drugs, that's not the case, and so they actually don't advance any further. If you can pass this stage, you can actually go into very large studies usually, and you want to show a statistical effect in a large population and a big benefit that is also safe. And very, very few drugs make it. So out of these 10,000, maybe 2 have that step. 60% success rate. So in the end, you maybe get 1 medicine out of this entire ordeal. And it takes, 10 years is optimistic. Usually it's more. It can be 15 years also in some cases. So it might feel a whole career for you to go from a very early idea all the way to helping a patient. And I think it's also very unique for an industry. So most projects, most developments that you would do will not take this amount of time. So drug discovery, a very, very unique domain in that sense as well. I think what you can take away about this entire process is it's really an information science. So it's really about trying to make the best decision at every stage, kind of anticipating what might happen in the future. Because if you're really good at selecting the one thing that works here, you can actually squeeze through all of this process, right? And you can maybe avoid having to do 90 other experiments that don't work. So if you have a really good predictive model, a really good understanding of the world, you can actually potentially really change the game here a lot. And this is really what we're after with these AI models. But it is still, I think, one of the most complex human endeavors to go after this. So in order to understand this kind of AI-driven stack for biological discovery, I think a really good thing is to look at this picture about how we can abstract biological discovery into kind of a loop. And really, this is just a formalization of the scientific method, where basically we're starting with, you can actually start in any point of this process, but let's say you're starting with some starting data, and you're really synthesizing that into some kind of understanding, so you might want to formulate a hypothesis based on that data. For example, you could say, for example, a gene is attributing and contributing to a certain disease, and that could be your idea, right? You could draw some connections between that, and you could say, okay, this is what I'm going to go after. You might take that into an experimental design, so you might say, okay, I'm going to set up a study to test this idea in the lab or in humans, whatever you're interested in. You would then run these experiments, and you would do some kind of measurements to find out, is that true or is it false? Based on what you generate in data, you find something about your initial hypothesis. This can again be synthesized, can lead to a new hypothesis or the same hypothesis in a different way. And so you can repeat that loop, and this goes on all the time. Interestingly and intriguingly, this actually captures the entirety of drug discovery. So from the earliest experiments in cells all the way to the human, where really this is a clinical trial. So in the end, really this framework really helps us understand drug discovery at large and how we can use machine learning to help. And so I'm inserting here some pictures where we can insert AI in this process, and these are all the topics that we will be talking about today. The first one here is one obvious way of changing and improving this process is you can skip the experiment, right? So if you can not do 2 years of an experiment and set it up with maybe $1 million investment, instead can have a computer compute the result of that potential experiment, that could really accelerate this process. You can run much faster, you can learn much faster about the world. So this is what we call in silico experimentation, and this is really what we will be talking about first. The second thing that we can then do is you can actually use that capability to also design your experiments better so that you go after the things that are most likely to succeed. And this is what we call optimal experimental design. That's the second thing we'll talk about. The third thing is actually synthesizing data. This is often an underappreciated step, but a lot of this data is coming out is not really useful. So you want to find out which of these data is actually useful, which of these data can you trust, and which of these data actually tells you something about the human. And for this, we use what we call causal discovery, and we will be talking about that a little bit in the third part. And if we get to it, I'm a little bit optimistic that we can, but maybe we have a little bit too much content. If we can't, I will send you the slides, of course. But you can also use machine learning to make more of that data and really impute some of these data that you maybe weren't able to collect, but maybe it's quite predictable from what you've seen in the past. So all of these things are things that we can do to improve this process and insert ourselves with AI methods to really improve what is going on in the practice. Now let's talk about perturbation. Well, let's take a quick break first. Do you have any questions about this? Is this clear, like the setup, why we're doing this, how this fits in? Any questions? No? Okay, let's go on. So the first thing that we want to do is we want to talk about doing these experiments in computers. And in order to do that, it's maybe good to understand first what are these experiments and how do they look in practice. And this is one nice example, is a real example from basically this experimental design. You're starting with some cells, so this is a cell type experiment, where you basically have some cells in a Petri dish, and you're basically usually doing the following. You're taking what we call a randomized controlled experiment, where basically you're taking one path where you're not doing anything with that, right? You're just leaving it as is, and then another path where you're basically changing something in the system. Usually that something is something that changes, modulates your process in a way that you think is informative to understanding what is going on. So you might be applying a CRISPR to change the genome of that cell, or you might be applying a small molecule to intervene in some way on some biological mechanism. You usually then, as a key step, you might do some kind of measurement. I'm using here the icon of the microscopy, so that measurement might be high content imaging, so you could literally just put a lens to that cell and look at what it looks like. And this is really what the example here gives as well on the left side. Left-hand plot here being the control, right-hand plot being the treated setting, and these are cardiomyocytes, so heart muscle cells, where the right side is treated with retinoic acid. And really, I think it makes very clear why this works. So you basically have here the difference between the two, which really illustrates what is the causal effect. And here it's very clear because we have the staining, of course. And so really what we're looking at is we're trying to understand the delta, the difference between treating and not treating, and this is what we call the causal effect. And we're doing this, you have to think about this not as a one-off thing, but actually as a thing that we can do hundreds, thousands, sometimes hundreds of thousands of times. In fact, we have robots doing this sometimes for us. And you can then think about on a meta level, right? You can take all of these experiments and you can put them in a space. You can say, let's say this is some kind of two-dimensional embedding of your experiments, where you're starting off with some, let's call it a starting state, maybe we call this an unhealthy state, and you're looking for something that moves your state, the delta, the arrow, that changes your state. You want it to go towards what we would call a healthy state, right? So something where ideally it changes, ameliorates the phenotype that you want to modulate. And you can then plot these arrows, right? So these differences in what is changing in the system, you can basically just attribute to that change vector. For example, it could be that small molecule 1, small molecule 2 that are really moving the state, but not quite to the healthy. And then you maybe have a CRISPR intervention that takes you that way, and then you can really use that as a really starting point for your next step in the medicine development, where you can say, okay, this is effective at moving me from that one state and to that goal state. And so really that could be a potential first idea to go to the next stage. So really think about this as a thing we can do not just once, but maybe hundreds, thousands of times, and really get all of that understanding of the space of interventions, the space of causal effects that gives us this kind of broad understanding of how we can change cells to go in the right space. Now, there's a lot of challenges actually using machine learning to try to predict these experiments. One of the problems is really well illustrated by this graph. So in the causality field, we often use what is called the directed acyclic graph, which is basically just a visualization of causal relationships in a kind of directed manner. So you have these nodes that correspond to entities in our biological domain that very often are genes, and collected with arrows, which might imply that there's a downstream effect, right? So if I modulate gene A, it will change gene B and gene C. That is indicated here by the arrows. And you might have a measurement that you're taking as well, which I call here readout Y, which indicates some kind of distribution that you're observing in this kind of network of causal connections. If you're then looking at the perturbed setting, so that's the control case that I showed you at the left side end of the experimental setup. On the right hand, you have the perturbation case. And in the perturbation case, when you have this kind of acyclic network, what you're doing is you're intervening on one node. Let's say you're doing a CRISPR-I, knocking out gene B. Then what you're doing is really you're setting expression, so all of its downstream effects to 0, and all of the incoming nodes don't matter anymore because we basically deleted that node from the system. So this is really what we're doing when we're doing these experiments, and this is kind of the abstract view of what that means in a kind of acyclic graph sense. What we're then observing is possibly a change in the readout, right? So we're measuring something potentially with these microscopes, and we're seeing then that this distribution that originally was this shape, it might move into this shape, and that might imply that there's some change downstream of this intervention, which might imply there's a cause effect there. This task, predicting these types of results, so these types of shifts from intervening on certain nodes, we call this counterfactual inference. It basically just means, what if I were to intervene on my system, what would happen? And it's really what we get out of experiments typically. Now, in order to use computer science and machine learning models on this is actually quite hard, because you can see the key issue here is that this is discontinuous, right? So you're actually completely changing the generative process, how the data is generated, in a discontinuous fashion, where most machine learning models, they assume a certain level of smoothness, so there can't be like discontinuous changes. So in order to be able to do that and use machine learning models to do that, we actually need to change how we do machine learning, and I will give you a few examples of that. So most models, they basically assume if you use like a multi-layer perceptron, right, and you're trying to model a setting like this, you're mostly just going to be able to model this control case. You're not going to be able to model this counterfactual case. Reason being, you're changing the generative system, so your model assumes it's going to be smoothly connected with this, but the reality is it's not. You're actually intervening on it, you're changing the process, then your model it doesn't know anymore how to extrapolate in that setting. So what you need to do is you need to introduce what we call a level of conditional plasticity to be able to model that conformational change, so the change between the generative process from the control case to the perturbed case. And we introduced a number of machine learning architectures to be able to do so. Basically, you just need to change your architectures so that they're able to extrapolate and to account for that change in the conformation of your setting. And you might be able to do that by conditionalizing, for example, and then having different paths for these downstream predictions for different counterfactual settings. That's one that we developed for doing multiple treatment perturbation predictions. We did another version that actually has a second level of that, because when you're doing this for different dosage levels, you also want to be able to understand what is the level of dose and the influence of that. So you can actually introduce on top of the treatment arms another arm that really gives you the ability to predict what is the effect then of different doses, for example, of different doses of small molecules applied. You can extend that even further to even be able to do that in combination. So as you can tell, there's a combinatorial explosion there, where basically you have infinite options to combine the different treatments and dosages thereof with each other. And so you need a model that's able to kind of account for that. And this is an architecture that we developed that is able to account for that conditional plasticity, even in the setting of multiple treatments that are used in combination with each other. Why we're doing this and why is this super helpful? Because as I told you before, in this kind of loop of biological discovery, you can actually insert then the model into your process of the experiment instead of the experiment. And what you can then do is you can basically just do the counterfactual prediction. What is the potential outcome of your experiment? And that you can then use to generate your new hypothesis, and you can rank them by what are the most interesting results for you. And then instead of doing all the experiments, you just do the ones that have the interesting results predicted. And then you basically get a higher enrichment of positive experiments using this type of approach. It's not always going to work. Obviously, the prediction model has some error associated with it. But if it just works more often than the random approach, then you basically get a higher efficiency out of the entire experimental exercise. And we tested exactly that. I won't bore you with all of the benchmarking, but basically we used that, and you can see accounting for this conditional plasticity in your machine learning model, so not just using the supervised learning, which doesn't account for the conformational change, but using it here in black and gray. You can see consistently across all the settings that we tested at, so these are all like counterfactual settings, both simulated, so when you know the ground truth graph setting of the system. Here you can compare with standard machine learning models that are supervised learning. You can see a clear improvement by the models that are able to account for conditional plasticity. Also for combination therapy, HIV, very interesting setting where basically the current state of the art is to treat with a lot of different treatments at the same time. You can see these models are also able to predict better the outcomes in that setting. Similarly, for another HIV cohort that we looked at, and if you have a three-way CRISPR knockout, so if you're doing an experiment with 3 different CRISPR combinations, you can also see that these models with conditional plasticity do a lot better in predicting these what-if outcomes. So that is a really effective and first instance issue that you need to address, which using these types of approaches you can really start to do. Now, a second, well, let's take a quick break with any questions. Yeah, go ahead. Sorry, can you?
So how is your approach? I think the method you proposed is called ENCORE, if I remember correctly. And my question was, how is it different than a TARNet? Because one of the benchmarks or the methods you compared it to is a TARNet, which also has these 2 treatment arms. So I was wondering, how is it different than a TARNet?
So the ENCORE is actually for a different setting. The TARNet that you know of is binary. So there's 1 treatment and 1 control, and only that. This model actually can predict any number of treatments and any combination thereof. So let's say you have 100 options to treat someone. You can pick 5 of them together and predict what is the result of that. With the target, you can only have 2 options, control and 1 perturbation, so you would have to develop hundreds of models to make the same prediction that this one model does. So it's a different setting. This is the multi-combination setting. The other one that I showed was the dose setting. Also, the target doesn't have any understanding of dose, so this is an extension into that setting. There's also a multi-treatment, so again, you can do 50 treatments if you want to. And then for this one, this is actually also multi-treatment, so the simplest variant, I guess, here, not with the dosage, is also more than the 2 arms. So basically, it's just accounting for the conditional plasticity in more complex settings, more realistic settings. So in the drug discovery setting, we rarely have only 1 option. In fact, usually it's infinite options, and usually it's combinatorial as well, so you can add multiple things together. So you really want these more complex settings. That's basically the motivation behind this.
So in the performance plot you showed us, how did you here compare it to the target if the target was not meant for the same setting?
I think what we did here is we basically just built 500 of them, right? So you can assume the others don't exist, and you can do one, and then you build a lot of them, just to be able to compare. It's not a fair comparison because they didn't build it for this, but it's the only way you can use it. And at the time when we wrote this, there actually wasn't any other alternative, so we wanted to at least compare to this one, even though it's not the same setting.
Okay, thank you.
Good question. Any more? Oh, there's another one.
Could you comment a little bit? I'm not familiar with the models you just presented. Could you quickly comment on the training mechanism, or sort of, the training regime for it?
Yes, I can quickly comment on this, but in the end, I think I have to refer you to the papers because it's just also a different way of training that you have to do, because in the supervised learning, you're also assuming that the frequency is basically the most meaningful thing. In the counterfactual setting, this can be very misleading. Just because something appears more often, that doesn't mean necessarily that it's more likely causal. So for these types of approaches, there's also what we call batch adjustment, where we basically try to make sure that things are not weighted by frequency, but rather by their counterfactual appearance. And so there's different training methodologies that we introduced in different types of these papers. So not all of them use the same approach, but in general, what you want to do is you want to account for this frequency bias so that you don't just see what is often happening, which has often the bias that we just measure what we can measure oftentimes. So certain technologies can only do certain things. You don't want to overweight on that. So basically, you want to adjust for that. And that's kind of the gist of what we're doing in these papers. And in the papers, you can see the maths and everything. So I just gave you the quick rundown.
All right. Yeah, I guess that will have to be enough for now for me. Thank you.
Cool. So we want to build on this and go to the next level. So one thing that I didn't tell you about yet, so we're increasing the complexity level even further throughout this presentation, is actually experiments are not static entities based on these configuration parameters of what I do, but also where I measure it. So what is the system, the biological system that I try to reflect? And I show you some examples here. So we already saw this example of dish experiments, but there's also what we call, for example, organoids, which are more collections of cells, and then could even go all the way to an animal model, in some cases even to the human, right? So we're doing the clinical trials to measure ultimately the cause effect in a human. Now, this is trivial if you think about it, but requires really some thought here in the machine learning sense, is that because of that, you might observe different things in different systems, and that might not be a reflection of your intervention not working. It might be a reflection of the system not being the right system to measure a certain thing, or just being very different than the other one that you're looking at. So you really want to have a systematic understanding which perturbations do which things in which type of system, so even upping the level of complexity a little bit more. And in order to do this, we developed recently what we call the Large Perturbation Model, LLPM, which basically is the first model that we've seen that can basically disentangle the axis of context from the perturbational effect in order to make a prediction that is context dependent and understand really which types of effects are happening in which types of system, given the amount of training data that we have today. And I will just quickly explain the principle. So let's say you have these experiments, which throughout this presentation, we indicate with these dishes, which basically just say this is the model system that we're looking at, these arrows, which basically say this is the perturbation, the intervention that I'm doing, and the microscope, which basically indicates the measurement that I'm taking. Now, all of these are different configurations, right? So you have different types of perturbations, you have different types of model systems, and you have different types of measuring, right? So you can measure the RNA sequencing, you can measure the proteome, you can measure the imaging, and we have tons of experiments, right? So I'm coloring here the different experiments with the different configurations in different colors. And you can imagine you have not just 3 of them, but really thousands, maybe millions. So these models, when we train them in the realistic setting, they usually have millions of experimental data points available. And you can really think about this model of a 3-dimensional structure, a cube basically, where each dimension it's trying to disentangle the contribution of each of these experiments in terms of the readout axis, the measurement, the perturbation axis, and the context axis. And you can place all of these points, right, the green one, into wherever it corresponds to into this space. And now you can already see from a machine learning point of view, once you have sufficient amount of data points to fill this cube, you can start to predict things that maybe you haven't yet done experimentally using the extrapolation capability of the machine learning model. So you basically train on this type of data with a very large representative data set to build what we call the large perturbation model that combines the understanding of all these measurements, all these model systems, and all these perturbations in an end-to-end system. This is quite cool because basically what you can then do is you can ask queries of this type as well. So you can say, I'm interested in perturbing, for example, STAT1 CRISPR-I intervention. I want to read out the gene expression of PSMA1, and I want to do that in an A549 cell. Probably most of you just won't tell anything, but it's just basically a very specific cell line. And then you get the outputs from the model, which basically are the counterfactual prediction that I just told you about before, right? So what happens if I run this configuration of experiment? What is my likely outcome? The other things that you get is an embedding of each of these axes, right? So it disentangles the contribution of all these systems. So you can take a 2-dimensional slice out of each of these cube sides, and you can basically then just say, okay, where is my perturbation in that? Where is my readout in that? And where's my context in that? And actually enables a huge amount of cool biological discovery tasks, and I'm just listing a few of them here, and I will show you a few of them throughout the slides. One is obviously that counterfactual prediction, so you can basically just predict what would be the outcome of an experiment that I haven't yet run that fits somewhere on the space to extrapolate. You can also ask questions using these embeddings, these 2-dimensional slices into this cube. You can ask, for example, my CRISPR-I perturbation here in blue, how similar is that to my small molecules that try to target this this particular pathway, and then you can use that to really validate your idea that this drug may be doing a certain thing, so really understanding the mechanism behind it. Molecular function, you can also associate certain perturbations with certain pathways, for example, nucleotide excision repair, and if you know where that is in the space, you can actually then say, okay, your new perturbation, how likely is it to be closely modulating that part of the function of the cell? And that's really super powerful. For most drugs, we actually don't really know what they do, so having this kind of data-driven way to be able to say that is really cool. Another thing you can do is you can use the C-axis, and this is quite powerful as well. In the general case, we actually don't know between these systems what is the contribution of them, how close are they to the human. But using a machine learning model like this, you can actually start to do that because you basically have the similarity metric in that space. So you can say that my cancer cell line and my organoid is close or it's far away, and depending on that, they might be reflecting the same mechanistic background based on the data that it has seen. And then lastly, you can also potentially use it to infer the interaction diagrams that I showed you before. So you can actually build bottom up from the experimental data what that might look like, and I will give you some examples throughout the next slides. So as I told you, it's kind of a question-answer mode. So you can basically just say, I'm interested in my in vitro cell. I'm interested in my CRISPR-I perturbation. I want to measure some high-content imaging, right? These images that I showed you. And I want to know what is red, what is green, right? So these kind of predictions. And you can do that at any length of time and any amount of experiments that you want. You can already see the power of that. Basically, it allows you to explore the space in a much more rapid fashion than you would normally. The typical experiment, it takes months to years, right? So we're having acceleration by a huge time factor here if we're using these models. First thing you probably want to know is, does this actually work? So we did some extensive benchmarking. This is LPM in blue compared to all the models that you probably have heard about that claim to be able to predict experiments, most of which pretty bad actually in terms of the performance when we look at this. These are, so at the x-axis you can see different settings. So the first line gives you the type of perturbation that we're trying to do. So CRISPR-I is a genetic intervention, but also the model that I've shown you, it can actually do any type of intervention because it's so general. So it has this very general perturbation space, and it can place any perturbation in it, including compounds, small molecules. So it can also predict the outcomes for that. And then the second line is the experimental context. So that might be, for example, RP1 cells or other types of cells here. So these are all different types of context and experimental configuration. Y-axis is the performance, so the correlation between what we know to be true from held out experiments and what the model predicts. And you can see here throughout all of these settings, basically, the LPM is able to do much, much better than other existing approaches. One of the reasons for that is this outperformance is because it learns the context from so many experiments. So you can obviously learn something about a new experiment from all the other experiments that you have done. And most of these methods are not able to integrate as this model is doing all the data from all of these different experiments. So this gives a huge performance boost in the end. But still, in absolute terms, obviously it's not perfect. So we're talking about a 60% Pearson correlation, which is quite nice, accelerates you, of course. But for being able to do that instantly, basically, is quite an achievement, I think. The other thing that we did is we looked at that also in the single-cell context. I won't bore you. Also in that context, the model performs much better. And then I wanted to show you a little bit of the qualitative data, because that's always the most exciting for us. We, as computer science, we always love numbers, but biologists and medicine experts, they really want to see that it makes sense. And for this, you can potentially look at plots like this one. So I showed you, right, this model, it basically has this disentanglement of these three axes, and you can then take an embedding out of each of them to basically place it in a two-dimensional space. And one thing that we wanted to know is just, does this make sense from a kind of cross-modality sense? So this plot is showing you different types of perturbations. So this is the p-axis, and you're basically looking at compounds, small molecules in the dot, and CRISPR-I genetic changes in the cross. And I've annotated them here. These are the targets of that CRISPR intervention that you can see, and they have the same color if there are compounds that are trying to target the same genetic change, inhibiting the same genetic change as the CRISPR-I is trying to do. So your expectation, if you're going into this as a biologist, you would hope that the inhibitors, they are somewhere around. You would hope that the inhibitors, they are somewhere around clustered around. Unfortunately, I've lost my battery, so I can't show you this anymore. Basically, the dots, they should be close to the crosses. So you should have some understanding that whatever is happening with your inhibitor, it should hopefully mirror what you're trying to do, which is target the same genetic change that you see in the CRISPR-I. Interestingly, so this is mostly true, right? So if you look at this plot in more detail, you can actually see that usually what happens is the inhibitors are somewhat offset of the genetic change. So there's a small shift, and it's usually all of the small molecules together, and then somewhere the genetic change close by, but not exactly the same space. And that's usually because small molecules, they're not as specific. So they do lots of things. Usually you can't really do the exact thing that the genetic change is introducing. So you would expect actually some kind of shift, and the model actually reflects that in its placement as well. Another thing that is quite interesting is I've indicated here also in the plot some statins. Statins, they're targeting HMGCR, which is here at the bottom. And you can see here two statins. Does it work here? Oh, thank you. So I've indicated here two statins that are actually far away. HMGCR is here at the bottom, and you would hope that the statins are somewhere close by, like this one, right? So you would hope that it actually does what the genetic change does. But you see here two that are suspiciously far away. And so first of all, you might think the model is wrong, but then we looked at what are these two statins. And these are actually the two statins that are very different from all the other statins. One is benfluorex, which you probably don't know, but had severe side effects, was ultimately withdrawn from the market for that reason, is placed by the model far away. And this is actually a good thing here, because you would have hoped that the model is able to find that this actually does a lot of other things that are potentially not doing exactly the same as this. And pravastatin is actually also an interesting one, because this one doesn't have safety effects, but it has a lot of known immune-related modulations as well. So it's also very different from the other statins. So it's very, very, very cool to see the model not only reflecting the things that we expect, but where it doesn't match what we expect, it actually is also meaningful. So from a biological perspective, this was really eye-opening plot. Another thing that we did is, I told you, right, you can try to map perturbations to their functions. And so same plot again, slice through the p-axis. And here I'm now coloring the plot, not by the genetic change that is introduced, but the pathway membership of these different perturbations. These are genetic perturbations. And you can see here exactly what you would expect. The model is able to identify that these are all members of the same pathway. So if you perturb them, they basically have the same downstream effects, similar, not always the same, but similar. So you would hope that they're close by to each other if they're in the same pathway. And lo and behold, this is exactly what the model learns, is that these perturbations are close by. So another qualitative validation that this model actually learned something biologically meaningful. And the model is not trained on any of these pathways, right? So it only has the experimental data available to rediscover that these are pathway members. Another thing that I told you about is you can use this to infer causal connections. And causal connections, I think one of the challenges we face typically is we want to understand the whole system, but we can't test the whole system. So in order to be able to do that, you can basically think about doing some experiments in red here. This is the readout axis, and this is the perturbation axis. You can only do some experiments. You're not going to be able to do all the experiments. But what you can do is you can use the computer model to fill in the rest, right? So all of the parts of the space that you weren't able to do because you don't have the budget, it's not experimentally possible, whatever, you can fill in with predictions. And you can then do both of the inferences, one without the other data, the predicted data, and then with the predicted data. And we tested whether or not it's able to enrich our understanding, our ability to recall causal connections with that enrichment. And here I'm giving one good example that really illustrates why this is powerful. This is a protein complex, the ribosomal proteins. Small ribosomal proteins. And so we know they should all be connected. They're basically part of a complex. They form a unit. And so you can see if you don't have the imputation, so if you have the limited experimental data, you see a lot of sparsity, right? So there's a lot of connections that are missing that should be there, that you can't infer because the experimental data is incomplete. Now, if you use the perturbation prediction and fill in the blanks, what you see is actually the picture gets much more complete. This is actually almost completely filled in all of the missing connections. So you can see the power of extrapolating and the power of filling in the blanks in the experimental sense using a model like this, also quite cool. Another thing I wanted to show you is, and this is really why we're so excited. Sorry. Good.
I'm trying to understand in some way where are we getting the information from here? Because sort of famously in machine learning, the extrapolation is hard, and I'm trying to understand where does the sort of predictive power of these models here come from? Some kind of, I don't know, combinatorial generalization type of thing, or?
Yeah, I think, sorry. Oops. I think a lot of the power comes from, if we look at the benchmarking, this is my hypothesis. I mean, I can just try to find an explanation. Of course, I haven't causally verified this, but in the end, what I hypothesize is that there's transfer learning. So if you have experiments on a lot of things in a lot of different systems, then some perturbations, they have generalizable effects. So you can learn from having seen this in one cell line to what it means in the other one. Similarly, obviously, there's some overlap between your organoid and your cell line. Not everything is overlapping, but something that you can learn. And basically, the model is able to learn from these other experiments what might be extrapolatable. So it's a form of interpolation, really, because if you look at this, this plot here, right, it's just basically you're looking at other points that are close by in the space, and you're filling in the blanks by extrapolation between those points. So really, what you're learning from is the other points. And because if you have a large coverage, which fortunately actually there's a huge amount of some of experiments, not everything is filled in good, but there are certain experiments, RNA sequencing, for example, cell lines where we have a ton of data. Like, it's almost a waste of time for you to ever do one of these experiments again, because we have so many that we can actually predict quite well what is happening in these systems. So depending on how well covered the space is, better the prediction. And you can see here, by the difference of all the other methods that are not able to do transfer learning, the fact that this one is and is performing much better, it implies that there is amount of transfer learning that you can do.
Yeah, that makes sense. I just kind of wanted to ask if you have any kind of pinpointed intuition as for what allows this to work. So
I attributed 100% to transfer learning. I think there's probably nothing else that contributes here. So I wanted to share also this, because this is kind of connecting all the dots. So This is actually something that you can do with these models. Is there's certain experiments that you could do that verify that potentially you have something that modifies a disease-related process. And there's an interesting disease called autosomal dominant polycystic kidney disease. It's a mouthful, ADPKD, which is one of the more common rare diseases. It's a key cause of end-stage renal disease, which is basically dialysis need, and something you really want to avoid having. But this is a really good disease to test here, because we know very well what is causing it. So basically, there's a really strong relationship between how much functional PKD1 you have and how healthy your kidney is in this polycystic sense. So if you have a higher amount of this PKD1, in the functional sense, you get basically a healthy kidney. If you have a lower amount of it, you get a polycystic kidney. The reason that we know this is genetic studies, so we have large amounts of people with different variations in that PKD1, and different dosages and strengths of it, and we know that the people that have more of the functional one, they basically have the more better outcomes in this sense. So we know really well what we need to modulate to do better for these people, and a therapeutic strategy, if you think about the setting, is can I find something that increases my PKD1, the functional one, in order to help these people, right? So the ones that maybe have by birth half of it functional, can we increase it by double, and then we would fix the problem, right? We don't need to do anything more than that. And so it's a very cool setting because we can define the mechanism quite well, so we know exactly what we're looking for. For the LPM, that is very cool, because if you know what you're looking for, you can just say, okay, give me all the things that modulate the PKD1 upwards. And for this, we actually filtered it further. We said, can you give me all the approved drugs that do that, right? So in that set of compounds that we trained the model on, there's a lot of approved drugs. There's medicines that are already medicines for other things, potentially. And you can see here, interestingly, some statins show up to be expected to be upregulating PKD1 in kidney cells, so we also asked the model to predict us in kidney cells specifically. And you can see here simvastatin is one. This is quite cool, and we were specifically looking for that because simvastatin is a very common statin that is given to a lot of people for cardiovascular prevention reasons. So there's actually a lot of people that have it already in real-world data. And so we were able to go back to another data set where we have patient records, where we have data about people that received simvastatin. And we basically just said, can we look at these people? Can we filter for the ones that had ADPKD? And can we follow them through? The ones that had that treatment with simvastatin in the real world and the ones that didn't. And then we basically just take time point zero, right, at the end of treatment. How well do they do? How well do the ones do that don't have this treatment? And lo and behold, you can actually, this is also accounting for all sort of confounding, so it's not just random analysis, but actually trying to really make sure that these cohorts are balanced. But you can see here, after balancing and after accounting for the confounding factors, you can see here people that have been exposed to simvastatin throughout their ADPKD journey, they progress to ESRD, so the end-stage renal disease, the dialysis stage, at a statistically significantly lower rate, and they progress at a slower rate throughout a long period of time. So from 5 years to 10 years, you have around 25 to 30% of people less going to the end-stage renal disease stage in a very large cohort in a statistically significant manner. Obviously, it's not an RCT, but it's the closest thing that we can do here. And it's quite cool that we can validate here from end to end the prediction of a model in a specific system for a specific readout that is genetically determined to find something that is potentially here a good candidate for treating. By the way, there's other evidence that speaks for this. In fact, there's a clinical trial going on right now with simvastatin, with another statin, I think, in an older population to test this thesis, not because of what we did, but that was already ongoing while we were doing this work. And there's a study, an RCT actually in young people, in juveniles, that was positive for some kidney markers as well with statins. So it's not completely random idea, but it's something where it's quite on the fence whether or not you can treat people with this and help. So our predictions and what we tested here, it speaks for this being a potential option for these patients, and something that we will publish quite soon, actually. I will pause here again just for any questions that you might have. No questions, very good. So yeah, I think this is a really good example where you can go all the way from a prediction to a potentially meaningful thing for patients, and I think this is really what we all set out to do when we want to develop these models. Now, we're going at a bit of a slow pace, so probably won't be able to go to all the topics that I wanted to talk to you about, but I wanted to also share this with you, which is really trying to go the next level also in terms of understanding not only the biology from the top down, right? So what is happening from the experiments, but can we also understand what is happening bottom up? So instead of only predicting what is the outcome of an experiment, can we also understand what is the process by which that happens? And for this, we developed what we call a generative model of biological mechanisms, trying to do whole organism mechanistic predictions. And the basic central ideas that I think I need to share you before I explain the model is there's one concept called the central dogma, which you've probably seen in a few talks already. I'm greatly simplifying it here, so if you're biologists, bear with me and don't get too angry. We're starting with the DNA gets transcribed into RNA, the RNA gets translated into a protein, the protein delivers some function, what we call a presentation, phenotype in a more complex sense, but presentation is a little bit more accessible term here. And so there's what we know to be a certain information flow in biology, where it goes from the DNA all the way to the function, and there are steps that it takes in between. This is quite an important concept. I will show you later why this is important. The other important concept is in order to study what is happening in people, we can use genetic data, right? So we can look at large amounts of people, and I showed you as one of the things that we know to be effective in drug discovery. We can look at a large amount of people. We can look at, do they develop a certain presentation, indicated here with some coloring, and you can look here A, B, and C, and then you can compare their genomes. You can see, okay, these people with that presentation, they have a more frequent appearance of this change because the genome cannot be changed post-birth. Well, not in the generalized way. You can basically say that there's a connection there in a quasi-experimental sense because this is a natural experiment. And the people B here that maybe have not the presentation don't have that change. So you can basically make an inference about what we call here a single nucleotide polymorphism. Natural variation in a single locus. Now, the next level of that, you can obviously tell the limitation of that, which is a single location in a genome is possibly not very meaningful, but this is the current state of the art. So if you do genetic study, usually you're doing this type of thing. Now, to put this into perspective, your genome spans somewhere around 3 billion base pairs. So translating that into a library, that's 6,000 books, each with 250 pages. And you're just looking at one letter in the current genetic paradigm. So obviously you're missing the whole, possibly your story if you're doing that. So obviously what we're trying to do here is trying to really make that more comprehensive while following the biological information flow. And the model that we developed we call FenFormer, which is really a genetic language model. So think about language models in natural language, which read like text. And so just literally treating genetic code as text and having a model read that. And so we built a multi-scale genetic language model that basically starts by taking a genetic sequence and reading segments of that genetic sequence in a kind of comprehensive fashion around what is called the transcription start site, and aggregating all the parts of that sequence information for the whole genome. So we take actually whole genome WGS information here and on single individuals, and we make these predictions across the entire genome. We then infer basically on the next step. As I told you, we're following the information flow of biology. So in the next step, we're actually doing an interim step where we're predicting expression and chromatin accessibility based on what we see in that individual person's genome. That is actually only an interim step. So basically, we then do another level of prediction where we're going to the next level where we want to know what the presentation is of these people. And we've combined this here, large biobank data, with also disease outcomes and other phenotypes and presentations that we see in these people. So this model actually can learn all the way from the sequence to the disease and really do a backpropagation all the way through. So it's really cool and completely unprecedented. There's no other model that I've seen ever that does this type of bottom-up mechanistic modeling of biology in an end-to-end differentiable manner. And the output then is basically a phenotypic presentation prediction. So you can basically ask it to predict based on what you're reading in that genome, in that whole genome, those 6,000 books, 250 pages, what do I predict to be happening as your organism in all the different contexts. So it's context-specific prediction as well. So it makes different predictions for the liver, the heart. So it really looks at the entire body and what is happening there before it tells you about the organism level presentations. This is quite cool because you can basically follow the path, right? So you can follow the information flow in biology to make mechanistic hypotheses about why certain things happen. And so there's 3 tasks that we looked at for this model. One is really drawing these paths, right? So you can try to, every time it makes a prediction, every time it says, okay, this person person, it has a certain risk of a certain thing. What was the path that was followed and why? You can draw that, and you can draw that for all sorts of diseases. And you can also use the prediction capability. We also tested that. And you can even do another thing that is quite cool. You can try to do what I call mechanistic subtyping. So the typical subtyping is top-down, right, presentation-based. If you have psoriasis and two people come up to a doctor with a rash, he will say, okay, you're both looking similar, so you're both having psoriasis. This model is actually very different. It would actually do bottom-up, so it would predict why does someone have psoriasis, and you can differentiate between different types of causes for rashes, which is quite cool using this model, and I'll show you some examples later. One thing that you always want to see when you look at these models, you want to, as I said, qualitatively make sure that it actually does something meaningful. And so the first thing that we did is we just literally looked at, does it tell us things that we already know? So the model itself, it doesn't have access to literature, it doesn't have access to biomolecular knowledge, it only has the genome information. So is it able to rediscover what we basically already know? One thing that we already know, psoriasis is big, well-known skin inflammatory disease. We would hope that the model predicts this, right? We would hope that the model says genome, skin cells, inflammation is psoriasis. And it actually does this exact thing, so this is quite cool to see. And it tells us that one of the key mechanisms involved in psoriasis is skin cells and inflammation in people with psoriasis. So super cool that it's able to do what we already know. Now, where we really opened our eyes is when it started to predict us things that we didn't have an explanation for. And this is a really cool finding that we had where psoriasis patients, you think of it as a skin disease, right? And this is maybe one of the limitations of having this kind of top-down interpretation of disease that we typically do in the medical field. Actually, if we look at the epidemiological data, we know also that people with psoriasis have a threefold higher risk of having chronic liver disease. And we don't know why. If you look at literature, there's actually only the knowledge that it happens. We have actually no idea why, because we typically think about it as a skin disease. Lo and behold, the so the generative modeling, generative biological mechanism capability of it actually predicts this to be one of the key changes that are happening in people with psoriasis based on their genetic changes, is liver cell and hepatocytes changes, and it calls out steatosis-related pathway in the selenoprotein, but also inflammation-related pathways in the spexin-mediated inflammation. And these are some findings that we found to be the topmost predicted in psoriasis. So something that maybe is a little bit unexpected, but gives us an explanation for something that we see in the real world that we hadn't had an explanation for. So really here, filling in the blanks using the generative capabilities, of course, to be validated, but very cool that it basically gives us predictions that match some observations that we don't have explanations for. We did this actually not just in one disease, we did this in a whole range of diseases. So if you read the paper, we have a whole host of these examples. So diseases where we know certain things happen, but we don't know why, and the model gives us a prediction that matches that observation to a good degree. Another thing that you might be curious about is just how, I told you this model, it predicts different presentations. You might be interested in how well it does so, and so we compared it to this kind of state-of-the-art approach of taking this SNP level information and combining this is called polygenic risk score. So you basically take different types of variations, you put them together, these SNP level predictions, and then you basically make a bigger model out of that. That's currently what people do in this field. And comparing that to this genetic language model approach that doesn't take a SNP level view, but actually a sequence level view and understands the mechanisms behind that, we find that it outperforms all of the categories of PRSs that are currently in use to a large degree. And it has some 80% settings in which using the FENformer really enriches those predictions significantly. And then if you look at the second plot, which I found even more interesting, is one of the big problems that you have in this genetic risk prediction is that a lot of the data is from European and Caucasian populations. So usually they reflect more the frequency of what happens in those populations rather than the causality. And because this model is actually introducing a bias, an inductive bias, it's saying you should only predict things that are mechanistically connected, right? So it's saying you need to have a change in the liver with that gene product before you can make a prediction about this. And because it has that filter, it is able to filter out all the stuff that is only correlated but not causal. And so you see when you test this model in a non-European ancestry, it does even better, almost double as good compared to the other models. So this inductive bias, trying to make sure that what you predict is not only correlation-based, but also has a causal mechanism behind it that is explainable, really it makes it even more powerful and more transportable than existing methods. And in that setting, it's almost 97% of the time that these predictions are enriching current systems. This is across a number of major diseases. I didn't explain that. Lastly, I told you about this example of doing this bottom-up subtyping, right? So typically we define people to be similar by their presentation, and as you can obviously tell, there's probably hundreds of ways you can get a rash. So if you just assume everybody that comes in with a rash has the same disease, it's obviously very limiting, and if you give them a treatment, probably some will not respond because they have the rash not for the same reason as the other person. And so you really want to know why is someone having a rash, not just only that they have a rash. And so this is psoriasis. This is a plot, two-dimensional, where basically every single dot is a whole genome. And we asked the Fan-Former model to read all of those whole genomes here. And there's a whole, half of these people are with psoriasis, half of these people are controls. And you can basically then start to plot the similarity of what the model predicts to be mechanisms involved in that person's disease. And you can basically then ask it to differentiate, and I've colored it here for you, the different parts of the clusters that basically are based on the model predictions, supposed to be similar mechanisms driving the disease. And now you can basically look at a number of data points to validate your intuition and what I claim here, that the model is able to differentiate people with different mechanisms. One thing you can look at, for example, is their comorbidity rates. So at what rate are these people getting different diseases that are close by but not exactly the same in these different clusters? That would implicate that there maybe are different mechanisms involved that also have effects on other types of the system. And so, for example, here you can look at dermatitis, seborrheic dermatitis, and type 1 diabetes, immune-related. And you can see here, I pick out two clusters here which are quite illustrative. The brown cluster here, which has a very high rate of dermatitis comorbidity, a very high, almost a third of the people here have it, and sebderm as well. You have almost a third here, and almost no type 1 diabetes, almost none, very minuscule amounts. And these are all statistically significant, indicated by the star. And then if you look at, for example, the red cluster here at the other end of the spectrum, you have a lot of people, much fewer people, less than half of dermatitis, maybe a third of the sebderm, but much more type 1 diabetes. So much more. This indicates that potentially that there is here a more immune-related type of rash, whereas here at the top is maybe a more skin-mediated type of rash. And so this is quite cool because it basically tells us that looking at people from their bottom-up perspective, not just from the top down, tells us something about the potential mechanisms that are going on, and potentially that could be a hypothesis to start treating that specific group different than the other one that maybe has a different underlying causality. So I'll pause here again because a lot of information. Any questions? No, very cool. So we did this for a lot of diseases actually, and all of them have the same pattern. So I'll just show you one example, but in the paper you can find this for more major diseases, and everywhere we found the same pattern. So it's quite cool that our hypothesis that this bottom-up modeling and causal understanding of the information flow is actually quite impactful here for making a better model of the diseases here. Cool. So we've talked a little bit about running experiments in silico, all the way from running dish experiments, different contexts of experiments, to even a whole organism type experiment where there's no equivalent, right? I couldn't go to the lab and change a genome, but I can use a machine learning model to make the prediction what would happen and understand the causal path from that. So really cool. Some of these experimental predictions nowadays, they're even going beyond what the lab can even do. So it is quite cool capability, I think, in this particular box of the process. I know we're actually a little bit slow here, so we will definitely not get to all of the points here, but maybe we can get to the experimental design next. And then I think, how much more time do we have after that? Half an hour more? Okay, so maybe we can do one more. I was a bit too optimistic with the content here. One thing I wanted to tell you about is also that we can use these capabilities to run experiments in computers to then prioritize what experiments we actually do. So this is really the major benefit here, right? So any experiment capacity you have, it's always going to be limited. You cannot do all the experiments, and you have to pick them really wisely. And this makes the difference between an effective drug discovery enterprise and one that is ineffective. If you can select and use your capacity in the wisest manner, this is really the key unlock that you can apply these methods for. And so we actually published a series of papers on this topic where really you're leveraging this predictive capability to first run the computer experiment and then run the real experiment based on the predictions, decide which experiments you should run in order to really clear up what are the best hypotheses in that space with the same amount of experimental budget. A key concept that you need to understand here is in traditional active learning, and why are we even looking at it, right? There's basically a field called active learning that is supposed to be good at that. But if you look at the existing field of active learning, there's really two main method approaches that exist. One is diversity seeking. So if you look at a space and you would say, for example, there are certain peaks in that space indicated by deep blue here. You usually want to find, in drug discovery, you want to find all the different mechanisms in a space that exist that move your phenotype the most, right? So these would be indicated in these deep regions, these different peaks. And as I showed you already a few examples today, there's usually different ways to get to the same result. So you can find different mountains here in that landscape of value. And in active learning, traditional active learning, there's two main approaches. One is diversity seeking, so trying to cover the space quite well so that you find all of the peaks, hopefully. The other one is value seeking. So you're basically trying to only find the highest value path part of the distribution, and these could be the stars here. And the method that we developed is an Bayesian active learning method, which basically is aiming to find all of the peaks. So it's a different type of combination. You're trying to trade off the diversity seeking with the coverage of all of the main mechanisms that are moving your phenotype in the main way. So your idea is really, if you look at these plots, as you do more experiments, you want to cover more of these modes, and then eventually, hopefully, you're doing this faster than the other methods. So you can see kind of indicated here what you want to see. And same amount here. You're actually not looking for the highest value. So the value-seeking method, if you just keep, once you have found this peak, if you keep sampling this peak, that will be the highest value thing that you can do, but it's not super useful anymore because you already found it. So you actually expect this method to not give you as much value because it is more interested in finding the different mechanisms behind it. And this is really a second step, right? Here, this is kind of the experimental design. So we inserted the model here, and we make predictions, and then we use these methods to find out of these predictions of experimental outcomes, which ones should I actually run? Which ones should I actually do based on this kind of trade-off between mode coverage and value? And we did actually a very extensive set of experiments, explain here very quickly the different dimensions. You can think about how large is my experiment, so you can have different, usually what is done is plate-based experiments, so you have maybe a plate of 16 experiments or a plate of 30 experiments, right? And so you can look at that as an element of variation. Another thing is, what is the task? What are you trying to find? What is the modulation, the biological change that you want to find? What are the methods that you use? And obviously there's our method, but there's a whole host of active learning methods that you want to compare to. You want to use different predictors, and for example, you could use the LPM that I showed you as an insertion here, and you want to use different types of underlying models. This gives a very high combinatorial space, and we ran all these experiments just to find out what actually works in the space, and we published this as a benchmark actually called GeneDisco for this discovery, genetic discovery task. And I will just show a few of these plots here where explain how it works. Basically, you have on the x-axis different experimental cycles. So every dot is one run through that cycle, right? So if we go back to this, one run through this is one dot. So you're doing this over and over again to find all of the moles that are ideally throughout the same budget. And so you have here more experiments being done, and you always have the same budget, right? So you can basically say all of these different methods, they have the same amount of experiments they can run, and then you can count how many things have they found. for all of the different experiments that you're looking at, how many of the important known contributors have been discovered throughout this campaign. And you can see here, if you employ the strategy that I showed you before, where you insert the machine learning model as a kind of predictive element and then do the selection based on this kind of value and mode seeking trade-off, you can find that we find with the same amount of experiments up to 16% here of the entire amount of potential findings versus some of the naive strategies which only achieve around a 4% coverage. So you can find possibly 4 times more impactful things with the same amount of budget using this strategy, which really motivates why we're interested in this. This is a T-cell CRISPR experiment for IL-2 production. I won't go into the mouse because we don't have the time, but we also benchmarked this from the simulation side, and also there we see that this trade-off between the mode seeking and the value seeking is really very beneficial element of this Bayesian strategy. We also benchmarked this in a whole host of experiments, so I gave you this plot before, but we actually did this again for many different experimental settings, and in all settings we find that this method, this type of trade-off, and applying this kind of predictive experimental design method really gives you a benefit through the bank, also compared to all the state-of-the-art active learning methods here. And roughly it's usually a doubling of the rate on average. So yeah, we quickly went through this experimental design point. So really checking also the box here of helping us run the right types of experiments. Another question that you, before I go to the next topic, maybe questions? No questions? Good. Let's move to the next one. Next topic is then on the side of the inference. So really trying to insert ourselves in the synthesizing data step. So once we have generated experimental data, what can we learn from that data? Not all of this data is useful. Not all of this data will tell us something about the mechanism behind disease, and it won't be causally robust. So how can we use that machine learning capability to really help us do that in a systematic manner that is validated to be provably better? And we've developed actually a series of methods that are designed to help us both know what works, but also new methods that use the learnings of what we know that works to develop new approaches of doing so. The first one I wanted to share is called Causal Bench, which is our benchmark for inferring causal relationships from perturbational single-cell data. This was work with Uri's lab in Stanford. And the basic setting is the following. You've already seen it, right? We have a control case, which basically looks at the unperturbed setting. And these are called observational samples, right? So these are samples that just tell us about what the structure in an untreated sense looks like. And then you have hundreds of thousands of samples of hundreds of different interventions where you're trying to change these systems, and you're then observing the outcomes for these different distributions across the board. And this creates your training data set, which you then want to work with to try to find that ultimate ground truth network. So really what we're trying to find is this connection diagram here, the ground truth connection diagram that in general we don't know. So for biology, we have no systematic knowledge about this. We don't really know what these networks look like. So we really want to learn that from these experimental settings. And then the algorithm actually is using all of that data to try to find this setting. This setting is actually quite hard, and there's three types of really big problems. The first one we call these slides I've stolen from Peter Spirits from a NeurIPS tutorial, where basically there's 3 types of main problems. Identifiability, which is really about can I even using that setting, and is it even possible that I can identify the causal relationship under standard assumptions from the setting. And this usually requires what you've seen here, right? You usually require some experimental setup. This is why we can't just observe people in large quantities in the real world. If it's not interventional, we can't learn a lot about the mechanisms, because frequency-based learning is not necessarily causality. So correlation is not causation. Is an important element of why we're doing experiments, why we're doing all of this effort of perturbations. Statistical, so can we actually infer? So have we enough samples? Do we have a large enough effect size? Can we find that relationship under the statistical lens? But also computational, you will find that for certain graphs of a certain size, it becomes actually a very complex computational problem. So imagine you have, for example, a genome, average human, 20,000 genes on average. So you might have a 20,000 large node set, and then obviously each of them can be connected with each other. So you have an exponentially large space, which becomes, if you test them all pairwise, a very, very large problem. And so there's a number of approaches to try to do this. PC algorithm from Spirits is actually a very, very traditional method to do this, but there's also different methods based on greedy search, co-expression-based methods, for example, GRN boost, but also newer thread of work where people are trying to combine various levels of deep learning and causal discovery together. And these are the types of methods that we then try to benchmark in this kind of causal bench setting. And there's really two different ways we can validate whether or not such networks are good. As I told you, we don't have the ground truth, so we actually don't know in general whether or not a network is true or the predicted network is false. But what we can do is we can basically do two things. We can look at what is known about certain relationships. So if there's already knowledge about a relationship in some standardized database, for example, pathways are quite well mapped, you can use these databases, and you would hope that a method that is better has more correlation with that, right? So you would hope that as I do more better method development, I hope that my connections are more likely to be recalled. These databases, they often depend on data from physical experiments. So really, do these genes actually interact in a physical sense? There's also protein-protein interaction data, protein complexes that can be used, and ChIP-seq as well, where you can basically look at biological plausibility as a kind of test that you can apply for your predicted interactions. And the second thing that you can do is basically just statistical. So you can look at, if I intervene on node A, can I observe a distribution change in B? And then that can be a statistical evidence for a connection being given. And so you can basically just a statistical test to find out. And ideally, if you want to just be comprehensive, you do both of these things, and then you find across all of these tests which of your predicted networks are best. And that's basically the approach that we've taken for what we call the causal bench benchmark. First thing I wanted to share with you is just some of these plots, and I will show you a few throughout the next few slides. The basic idea is it's kind of x-axis true positive, so how many of the predicted edges were actually given then in the data source that you looked at, so the different types of evidence, statistical or biological informed, and then the true positive rate. So out of the predictions, how many of them were true and how many of them were not given in the database. So ideally, you want to have top right, right? Top right is the ideal outcome here. The closer you are to the bottom left, the worse your method is doing. And we benchmarked all the methods that I just told you about, including random selection. And you can see here that by and large, across all of these tests, most methods do relatively poorly, I would say, in recalling both biologically known interactions, but also statistically validated interactions on the data. So most of these methods that we see today, they're actually not that good, but out of the ones that we know, GRN boost did the best on average if you look at the rank across all of these different tests. And we do that also, by the way, in different settings. So RPE1, for example, is one of the settings that we looked at. Another thing that was quite curious to us is you would expect, by the theory, you would expect that you do more perturbations, you get better recall of the underlying ground truth. When we did this benchmark, we actually found that this is not true for most methods today. In fact, we didn't find any method today that scales with the perturbations, that gets better as you give it more perturbational data. So all of the existing methods, they don't scale with perturbation. This is very concerning because basically means you could collect any amount of data, you wouldn't get better understanding of biology. So this is quite concerning to us and really highlighted for us a problem in the literature where we don't have current methods that actually do better systematically as we do more data collection of perturbation experiments. So this was actually the starting point for what we called the causal bench challenge, where we actually asked the community, you guys, to develop us methods that can do better in this benchmark. And we awarded a prize, GSK awarded a prize to the winners of the challenge some 2 years ago, where Columbia, Michigan, and ETH Zurich were some of the top scoring solutions. And what was notable, if you look at the plots that I showed you before, where basically x-axis is fraction of how many perturbational samples you give the algorithm, y-axis is statistical statistical test results, so how many more of the statistical known relations were recalled. And you can see here the bottom approaches. These are the state-of-the-art baselines that don't scale. You can see that the new methods that were developed in this challenge, they actually do scale. So you can see here significant improvements as we get more and more perturbational data. So this was a completely new finding that really enabled us to move the field forward here in order to be able to actually leverage the now existing large-scale perturbational data to do much better at this task. So a huge contribution here by the community to do better. The second thing we're interested in is why do these new methods work and why didn't the old ones work? So we actually did quite a lot of theory work following this challenge where we just wanted to understand why the hell was this not the case before? What was the problem that we had in our past method development that we didn't scale with more perturbations? And this actually led to the development. So actually all of these methods that did well, they had similar-ish ideas. And so basically the concept that we were able to develop based on the learnings from that challenge were what we call epsilon interventional faithfulness, which basically just encodes this core intuition where if I intervene on something at the top, all of the children should be changed, right? So if I look at graphical relationship, I have here a key root node, and it's connected to 3 children, and then there's some downstream children as well, right? There's a possibly very long chain. You would hope that all of the things downstream have some interventional change if you measure their gene expression levels. And so you would hope that any children and this parent-children relationship, what we call causal order, so top things are below ranked higher, that is reflected. And this is kind of a key assumption that we introduced is that your data set needs to basically reflect that in a kind of epsilon thresholded fashion. So up to a certain confidence level, you need to be able to see that change. If you introduce this theory, then you can start to really get guarantees in this setting, scalable guarantees that you do better with more samples. And we developed a whole host of methods that leverage this principle of epsilon interventional faithfulness to develop scalable methods that leverage this principle in terms of growing with perturbation data. And we were able to show actually a theoretical guarantee that you would get closer to the ground truth. So it's really magical because in the causal world, it's really impossible to get any guarantees, basically. But under this assumption that you see the changes for the children, all the children, in the causal order sense, you can actually get theoretical guarantees and a convergence error bound even for this, which was quite eye-opening for us. And I can show you some examples from the experimental data. You can see here x-axis again, samples that you collect, fraction of the data set that you explored, and error is on the y-axis. You can see here, if you leverage this theory of the epsilon interventional faithfulness, you can give a maximum error bound, which you can see here. And then we developed a certain set of methods here, the bottom legend, where they were trying to leverage this principle of epsilon interventional faithfulness. You can see they all stay consistently below the maximum error that is theoretically predicted, but also that scales with the number of experiments that you have. So really, really nice that the theory here explains the practice, and we were able to develop a method that leverages those principles. And this is just a setting, just to explain it again, right? You have some experiments, variables of genes, you observe certain changes, and you can then derive this causal order with these theoretical guarantees in that assumption of the epsilon, under the assumption of the epsilon interventional faithfulness, which is a really completely new theoretical framework for looking at perturbation data in this setting. So quite cool that you can actually, in this setting, without having access to ground truth, get guarantees of being able to find out what nodes are upstream of downstream nodes. And this is really quite nicely validated in this experimental setting. Now the last section I wanted to talk you through is you can actually try to, once you have generated experimental data, well, first maybe I'll just pause because maybe there's questions about the previous section. No questions. Okay, let's go on. For the last part, I wanted to talk a little bit about how we can make more out of the experimental data that we generate. So we can, as I said, only generate a fixed amount of experimental data sets. We have a capacity, we have a budget. So how can we actually make more of that data by basically potentially inferring some things from other things that we already have seen in the past? And this is again using the setting of imaging experiments. So basically I explain again a setting with cell cell experiments where we're introducing some change, and we have a certain amount of a certain system that we're testing that in. And this is kind of the path that is fast and cheap, right? So we can basically just put a lens to it, maybe use some stains as well, so we get some coloring, and we just do microscopy with high content imaging, generates these nice looking images where we can see the cell and what the effect is of the perturbation from that. Now the more expensive part is we can basically look at, for example, mass spectrometry, doing proteomics, so really trying to measure the abundance of certain proteins. Likewise with the transcriptomics, we can do experiments. Depending on which experiment you do with which machine, this can be very, very expensive and can take very, very long. Whereas here at the top, this is very cheap. So we're talking about cents and minutes versus months and millions at the bottom here. So really what you want to do is when you have collected a lot of this paired data, which fortunately we have, so we have a lot of data where we have collected all 3 of these, you want to use the top one to predict the other ones, and you want to know that whenever you're going this fast and slow path, that you're not able to actually predict that a priori without having measured this before. And so if you have this paired data, you can actually start to potentially from the image predict what might be these measurements that you could take in a more expensive path. And in order to do this, basically we did the following. So you can look at these images and the different stainings, the different image stains that you can see here colored. And you can basically develop a machine learning pipeline where you're taking these images, you're tiling them in a cell-centered way, so you're taking the nuclei and you're putting them in the center of the picture. And then you're training a machine learning model using different loss functions to basically build an embedding space that reflects the similarity of the different images to each other. And I can take on top of that, train a machine learning model that just predicts you the different abundance levels of proteins, of transcripts from the image. And you can train it on a large data set, such as the one that we used for this paper. And we call this method image-tomics, where basically it is able to predict for these images what might be the corresponding measurements that you could take in the more expensive path. We tried that and we tested that systematically across all of the measurements that were available. And I give here some examples, for example, HLA-A, and on the right side some example images, cell-centered, and some attribution maps that the model focuses on. And then we looked at that also on these pie charts here systematically across all of the different measurements that we can take. And you can find here roughly some 10-ish to 15-ish% of abundance measurements were confidently significantly predictable from the images. And then there's a large part that is low to no expression, so the experiments actually didn't work in the bottom part, right? We didn't get a good level. And then a lot of it is also roughly 40% here are not predictable significantly from the image. So it shows that basically there are certain measurements that you can take that are predictable from the images. And we were also interested in finding out why and which types of features are indicating predictability of measurements. And one thing that we found to be quite well associated is abundance, which is not too unexpected, right? So if something is more expressed and more available in the cell, it has a larger effect on the visual of the cell. So we see that more expressed gene products, they're also 4 times more predictable than the lower expressed ones. You also see certain pathways being more predictable, but you also see certain pathways being less predictable. This is also somewhat expected, right? There are certain pathways that are having a lot of effect on the visual phenotype of the cell, whereas others, maybe they're not really changing the visuals of the cell, and so you can't really read it out of the image, and those are then enriched for lower predictability, and there are certain pathways that we found for each of the measurements. This is for transcripts, but we have all of the different plots here for the different measurements that you can look at. Another thing that the model learns implicitly, I've showed you this a few times actually throughout the presentation, but basically you can look at the model and what it learns in an embedding sense. So you can take, again, a slice out of the model, and you can just put it into a 2-dimensional space. And in this plot here, every single dot is a cell tile set, and it corresponds to the different settings. These are different types of macrophages that we had in this experiment, and there are different perturbations applied to them as well. And so this was kind of a broad coverage of biology, and this is why this is quite a nice training set. And you can see here the model learns to really differentiate the biological states in terms of the images, so it really learns the functional relationship between the image and the cell state. And this is ultimately what it then uses to differentiate between the expression levels and the gene products in the different omics layers. So again, this plot really validates quite nicely that what the model learns here is related to the function, and that is ultimately what predicts the omics levels using that kind of reference data here. So nice example of how we can use machine learning to really enrich our pool of data that we have available and make inferences here in a faster and much cheaper fashion based on scalable imaging technologies. So I think that completes our cycle here, right? So we went from all the way from doing faster experiments, predicting all of these experiments in computers, really accelerating this cycle, to using that data and those predictions to then make the most out of the fewer experiments that we should run, and then taking that data in and making sure we learn all of the things that we can learn from that data in a causal sense and discard the rest using causal discovery. And then lastly, how can we use those data points that we trust to make predictions about what else we can learn from that data that might be already given by what we've learned in the past about the relationships here. And so you can already see this picture kind of forming, right? So throughout the session that we had here, I think you got a nice view of what we imagine to be the future of biological discovery, where you're accelerating the process by basically giving every single biologist a little robot that has access to these tools and really allowing them to do whatever idea they have much, much faster, much, much more efficient, and not having to go through the whole cycle of doing experiments, failing, taking a long time, but rather just spending your time on the key things, the few key things that matter, and not wasting your time on things that don't. And the tools that I developed and that we developed in our team here, they can really help us do that and help us explore the space and the biology much, much faster in order to bring more therapeutics to patients with that causal evidence, which is the thing that we started out to try to do here today. To summarize, I just want to give you a quick view of what that means, right? So when we're doing this, even small changes can make a huge difference, and I've shown you a plot here that really illustrates that. If you look at the R&D drug discovery field in general, this is the amount of projects that exist. So these are the amount of, in the whole world, how many drug projects are running today. And we're looking at somewhere around, I think, 8,000, something like this, here in 2019. And if we use the historical success rate, right, we use the historical success rate of 5%, that would translate to somewhere around these few hundreds of drugs coming to patients. Now, just assume the tools that I showed you today, they just improve the process by 1%. 1%, nothing more. I'm not asking for a lot. If you do that, you get 80 more medicines. 80 more medicines for 1%. Now, my hope is we can do a lot better than 1%, but if we can only do 1%, we get 80 medicines that will change the lives of so many patients. So I think there's a magical opportunity here to improve the world, improve the outcomes for patients using these tools, and you've seen a few ways here in how we can do so. So I'm really excited about this, and I hope you've all been inspired throughout the summer school here to dedicate yourself to this topic, because there's a huge opportunity to help your friends, your family, yourself in the end as a patient. So with that, I want to thank you and for the attention, and also the team and all of the people behind the work. You can imagine it takes an army to do all these things, not just at GSK, but also all of our collaborators, which I've done a bad job calling out throughout the presentation, but I want to do here again. We worked with our colleagues at Oxford, OTML, Seifert, TU Munich, Stefan, the Intelligent Systems Lab by Schulkopf, and our friends at Cambridge as well, and Harvard Medical School, who helped us with the FenFormer model as well. So you can see quite a lot of amount of people needed to do all these things. And yeah, if you're interested in this topic and you're excited about this field, do consider working in the industry. We have openings for AI, ML roles, software engineers, internships, really everything. So have a look at gsk.ai, which is our website where we advertise these roles. And there's a lot of science behind this, so if you're interested, feel free to look into the papers as well. So thank you so much.
Thank you very much for a very interesting talk. And okay, first question.
So I have a question regarding the multimodal prediction of that omics data. So you showed that the one dot is multiple cells. So are you aggregating the single cell patches into a single prediction? So that's the first question. The second, on the image, if you cut out the centered cells, the neighboring cells also are visible. Isn't the model biasing towards them as well?
Good questions. So for the predictions, it actually uses a stack of tiled images. This is because when you take a cell experiment, you get a huge image, so quite big. So doing the cell-centered tiling, it helps the model basically focus on the cell states rather than counting things like how many cells are in that experiment, right? So really helping you bias more towards the cell phenotype rather than other metadata of the experiment, which often is also predictive, but not in a causal sense, right? So you don't want to find just that all the cells die. It's not usually meaningful. So we're kind of interested in more the phenotypic connection, and that's why we did the cell centering. You can see that doesn't always work. So there's certain experiments where there's multiple cells in the same picture. That's kind of a byproduct of doing it this way. But because you have the tiling as a certain size, it's limited the amount of things that you can learn in terms of the metadata from it. So yeah, but you can see also if you look at some of these examples, it does pay attention to how many cells there are in the different cells. So there's a certain amount of bias, I would say, is certainly probably there, but we haven't found a better way of reducing this yet. But if you have some ideas, definitely the field is open. We published this data set, by the way, so if you want to just build something on it, feel free.
Okay, thank you.
Thank you. Any other questions?
Thank you very much for the very insightful talk. I was actually mentally still stuck at the step number 1, related to the question that was asked before on the predictive power. I was wondering, because we're looking at all these dimensions, the dimension of perturbations, the dimension of the context, creating this cube you showed, I was wondering, isn't it super difficult to make sure that there is enough coverage by the data to cover this cube? I know we can impute, but we need to have at least all, let's say, dimensions covered by at least one experiment, right? I'm thinking here about the positivity assumption or the overlap assumption.
Yeah, great question. Certainly, I think there's spaces that are better covered. So as I said, RNA sequence is more common, proteomics less common, more complex measurements less common, cell lines much more common than in vitro organoid type experiments, animals even less common, humans very sparse in total, right? So for each of these systems, you have different levels of coverage that influences your predictability, that influences how confident you are. One cool thing that you get out of this model is you can obviously also calculate the uncertainty, right? And so this is something that when we use these models in practice, we also look at. And so generally, you wouldn't put as much faith into things that don't have a lot of backing data. So it's not magic, right? So obviously, it's based on data and it's based on what we have. And certain things we don't measure at all, right? Certain things we even don't maybe know that they exist. So we can't do experiments on them. But I think one thing that is quite cool about this area, and I think maybe where a lot of people get stuck, is that you don't have to be perfect to be useful. As I told you, around 1%, not more, you already get 80 new medicines. So maybe it's good enough to not be perfect, and if you have just accelerated, I don't know, 70% of the experiments by a lot, and avoided some experiments that we didn't need to do, that already helps quite a bit in the process that is overall quite hard. So yeah, I would say that it's a step on a staircase that leads to better and better models. But we were surprised already. If you ask me, right, I'm doing this for a number of years now. Generally, I'm surprised by how much we were able to achieve already in this amount of time. So I didn't expect this quick of progress. I didn't expect this many tools to be useful today. I actually thought this would be a decade-long journey. So I would say I'm surprised by how good it is, even though it's not perfect. And I completely acknowledge the limitations. And all of these are predictive models, right? So you should not expect them to be perfect. And all of the plots, all the benchmarking, we spend a lot of time on the benchmarking because of that, right? We need to build the level of confidence. We always check the quantitative, but also the qualitative. Does it learn something meaningful? And you get here the examples that work, but we have a whole trash bin of tools that we tried to develop that didn't have that. So you're seeing here only the best examples. So I would definitely say that is important, and don't take away that every model works, or that these are certainly the ones that pass that filter and that actually have the practical impact on that side. But there's 10 times more that didn't, that were maybe also had a good idea behind them.
Yeah, the great results you showed definitely show that the data you had was at least rich enough to achieve something. So is the data your company's data, or is it publicly available data?
What you see in these slides is published in the paper that is linked here. And it will actually come out quite soon. It's to appear, but we have a preprint out as well that you can look at. But the version that we publish here is all trained on public data.
Okay.
We have, of course, more data internally, but we're not able to publish that.
Thank you.
Thank you. Oh, okay. One more question.
Hello, thanks for the talk. My question is actually a follow-up on the data discussion. All of these causal methods that you train, which are, I would say, a bit different from the supervised methods that we have mostly, how are they performing compared to supervised methods in terms of this noisy public data? Because often with the supervised methods, what we see when you actually train them on a good internal data, they would really be much more performant. Are these causal methods, are they more robust to the noise that we have in public data, or do you see same or even higher jump when you train them on your internal data?
I think it's an emerging direction. I would certainly say a lot of the data that we do see in the field is pretty crappy, and you shouldn't probably trust it too much. But I think a model like this can ultimately in the future maybe help with that. I haven't tested, so it's just speculation, but if you train a model like this on a very large comprehensive data set that encapsulates a lot of experiments, then I think one thing you should pay attention to is if there's a very big outlier compared to what your model predicts. Because if your model predicts and it's a good covered space, to the comment before, right, and you have a lot of experiments like that and you did a new one that is very different, it's an outlier. If you have looked at a lot of other experiments and the model has seen a lot of experiments and doesn't predict that, I would be careful trusting that experiment. And so I would say maybe that's a way you can use these models in the future to maybe give a quality control to your experiments, because quite frankly, a lot of computer science, I don't know how many of you are computer scientists versus biologists, but if you're from the computer science field, you usually glorified experiments as ground truth. The truth is experimental stuff is as often wrong as the computational stuff. So you need to be very careful in trusting it all the time. And generally, you can maybe use sometimes experiments and a model that has seen a comprehensive set of experiments to validate whether or not the experiment has been successful.